DNA strand, that is template strand, from this mRNA is formed. From this mRNA, polypeptide chain is formed. First step, that is the transcription, and the second step, that is the translation. Amino acid sequence in a polypeptide chain is depend on the nitrogen base sequence present on the mRNA or nitrogen base sequence present on the DNA. This relationship is called genetic code. The relationship between amino acid sequence in a polypeptide chain and nitrogen base sequence present on the mRNA or DNA called genetic code. The relationship between amino acid sequence in a polypeptide chain and nitrogen base sequence in mRNA or nitrogen base sequence in DNA called genetic code. That means a group of nucleotides that represent one amino acid. So, genetic information received by RNA from the DNA is called mRNA. On mRNA, the genetic information present in the form of codons. These codons recognize specific amino acids. How many nucleotides that recognize one amino acid? Only 4 types of nucleotides present in the RNA. That is adenine, guanine, cytosine, and uracil. What is codon? A group of nucleotides recognize one amino acid is called codon. How many nucleotides or how many nitrogen bases? A nucleotide depends on the type of nitrogen base in RNA. Suppose if one nucleotide recognizes one amino acid, possible number of codons. A codon with one nucleotide or one type of nitrogen base. Singlet codons. Singlet codon. Possible number of singlet codons. That is 4 to the power of 1. That is a 4. Only one type. A singlet codon with only one type of nitrogen base. Only 4 type of nitrogen bases. So, only 4 types of codons are formed. So, total types of singlet codons 4. Suppose if you take the 2 nitrogen bases to recognize one amino acid, that is called doublet codon. That is 16. Total types of amino acids involved in protein synthesis 20 types. But the 16 types of codons not sufficient to recognize 20 types of amino acids. Suppose if we take the 3 nitrogen bases, that is called triplet codon. (64 codons that are formed), that is enough to recognize 20 types of amino acids. A group of nucleotides recognize one amino acid called codon. If a codon consists only one nucleotide called singlet codon, number of singlet codons only 4, not sufficient to recognize 20 types of amino acids involved in protein synthesis. If we take the 2 nucleotides, the 2 nitrogen bases, that is called doublet codon. Total types of doublet codons only 16. That number also not sufficient to recognize 20 types of amino acids. If we take the 3 nitrogen bases, that is called triplet codon, and possible number of the triplet codon 64, that is sufficient to recognize 20 types of amino acids. Codon is a triplet codon first stated by a physicist, that is George Gamow. Codon is a triplet codon first stated by a physicist, that is George Gamow. 24 codons, that is sufficient to recognize 20 types of amino acids. Now, total types of codon. Total number of codons 64. Out of 64, 3 codons not recognize any amino acids. They are called nonsense codons. Three. Number of sense codons, that is the 61. So, total types of codons recognize 20 types of amino acids which are involved in protein synthesis, 61 only. And types of codons not recognize any amino acids, 3. They are called nonsense codons or chain terminating codons. Nonsense codons or chain terminating codons. They are UAA, UAG, UGA. UAA, UAG, UGA are nonsense codons or chain terminating codons. UAA, opal, UAG, amber, UAA, ochre, UAG, amber, UGA, opal or umber. The name of that codons, nonsense codons, those codons are not recognized any amino acid, generally present at the termination of the polypeptide chain. So, they are also called chain terminating codons. UAA also called ochre, UAG also called amber, UGA also called opal or umber. They are present at the termination point, termination of the polypeptide chain. 61 codons recognize 20 types of amino acids. Here there is no any collinearity that we observed in the DNA strand. Suppose if we know the DNA sequence, nitrogen base sequence of DNA strand, it is possible to understand the nitrogen base sequence present on the mRNA. But it is not possible to recognize amino acid sequence in a polypeptide chain based on the nitrogen base sequence present on the either mRNA or DNA. But the sequence of amino acid 100% depend on the sequence present on the mRNA or DNA strand. The sequence of amino acids in a polypeptide chain depends on nitrogen base sequence present on the mRNA or nitrogen base sequence present on DNA. But it is not possible which type of sequence that recognize which type of amino acid. But it was clarified by various scientists. Haragovinda Khorana, he used the copolymers and homopolymers and solved the problems partially. Nirenberg, cell-free protein synthesis that deciphered this genetic code. Wachavo enzyme, that is template independent sequence of nucleotides that clarify this genetic code. polynucleotide phosphorylase enzyme, that is called Wachao enzyme. Next, properties of genetic code. Properties of genetic code. Genetic code is unidirectional. Genetic code is triplet. Genetic code is commaless. Genetic code is non-ambiguous. Genetic code is universal. Unidirectional. Starting codon is always AUG and termination codon is chain terminating codons UAA, UAG, UGA. So unidirectional, in one direction only the protein that synthesized, that is the 5 to 3 end direction. Genetic code is a triplet because there are 3 nitrogen bases. For example, UUU recognized as phenylalanine, GGG that recognized as glycine, GGG that recognized. That means a genetic code is a triplet codon. This triplet codon, one triplet codon recognizes one type of amino acid. Next, code is commaless. Code is commaless. Suppose on the mRNA, this sequence is present AUG, GUG, GGG, CCCC, like this one. In between any 2 codons, there is any punctuations, any stop signals. No letter is wasted in between any codon. So code is commaless. Code is commaless. In between any 2 codons, no letter or no nitrogen base or no nucleated wastes. So one codon ends with the next another codon, start another codon. No punctuation, no stop signals, no wastage of any nitrogen base. So code is commaless. Code is non-ambiguous. Suppose UUU that recognize phenylalanine. If phenylalanine is absent in the cytoplasm, that codon not recognize any other amino acid. So one codon not recognize any another amino acid. If that particular amino acid is absent, it never recognize another amino acid. So there is no any doubt. One codon recognize one amino acid only. Code is universal. Code is universal. UUU that recognize in phenylalanine in prokaryote, in bacterium also, in man also, in plant also, in any organism also. UUU that recognize phenylalanine only. That means one codon recognizes one type of amino acid in any organism only. But some exceptional cases, very few exceptional cases. For example, AGG, AGA. These two are recognized arginine. These two codons recognize arginine in all organisms. But in mitochondria, human mitochondria, they act as stop signals. That means nonsense codons. In all organisms, AGG, AGA recognize arginine, but these are the nonsense codons or the stop signals in case of human mitochondria. It is one of the exceptional cases. And stop signals like UAA and UGA, they recognize glutamate. In case of some ciliates, protozoans, So these are the some exceptional cases, but in most of the organisms, one codon recognizes one type of amino acid only. So these are the exceptions. AGG, AGA, these are the codons recognized arginine in all organisms, but in human mitochondria, they act as nonsense codons or stop signals or chain terminating codons. UAA, UGA, these are the chain terminating codons in almost all organisms, but in some ciliates like paramecium, they recognize glutamic acid, the glutamate. These are the exceptions. Next, code is degenerate. Code is degenerate. Here the 61 codons, sense codons, recognize 20 types of amino acids. That means one codon recognizes one amino acid only, but one amino acid may be recognized by more than one codon. A group of codons or two or more than two codons recognize one amino acid called degenerate codons. For example, serine recognized by 6 codons. Those 6 codons collectively called as degenerate codons. Degenerate codons, 2 or more codons recognize one amino acid called degenerate codons. Example, serine recognized by 6 codons. Those 6 codons collectively called as degenerate codons. For example, here, arginine, AGG, AGA. So, arginine recognized by more than one codon. So, these two are also degenerate codons. Chain initiation codon, generally AUG, that recognize methionine. Chain terminating codons UAA, UAG, UGA, also called nonsense codons. So, by the experiments carried out by different scientists, and it was confirmed that code is a triplet codon. Consider 3 nitrogen bases. Any change in one nitrogen base pair, changes occur in that organism. That type of changes generally we call as the mutations. Mutations caused by changes in one nitrogen base pair called point mutations. Best example of the point mutation, sickle cell anemia. Sickle cell anemia. In human hemoglobin consists the 4 types of polynucleotide chains, polypeptide chains. Out of these 2, alpha type 2, beta type 2, total 4 polypeptide chains present. In the beta type, beta globin, glutamate change into valine. Glutamate change into valine in beta type. Glutamate change into valine. So that cause sickle cell anemia. Codons that recognize glutamic acid or glutamate, GAG, GAA. Only one base pair changes, that is GUG, GUA. So here only one base changes. One base changes, automatically amino acid also changes. So sequence of polypeptide chain also changes. When sequence of polypeptide chain that changes, automatically sequence of amino acid in a polypeptide changes, type of polypeptide chain or type of protein also changes. So structure that changes, that leads to the sickle cell anemia. So that comes under the point mutation. What are point mutation? The mutation are the change that occur in one base pair. Suppose in this double stranded A that changes U, automatically in the complementary strand there is a T that also changes, another complementary that is collectively called as one base pair. So any change in one base pair comes under the point mutation. Best example point mutation, sickle cell anemia, RBC that becomes sickle shape, hemoglobin in that changes generally observed in especially in the hemoglobin, beta type polypeptide chain sequence changes. There is a beta glob change in beta globin. Glutamate changes into valine. So such type of mutations, frame shift mutations.